you remember, I was drawing out the big kind of sketch of cell-to-cell of animal virus replication. And so, of course, the first thing that we see is attachment as one of the first steps. But remember, almost in all viral infections, this is through some sort of receptor. So we call it receptor mediated. This can also be called adsorption. Not absorption with a B, but a D, which basically means the same thing. Grabbing hold of and sticking. And then with animal viruses, of course, the second thing is penetration. Remember, this is through that invagination and kind of actively endocytoses the virus into the cell. Then once the virus is in the cell, we have to get rid of that capsid coat. So we end up doing this uncoating. And what this does is this releases whatever nucleic acid there is. So if it's DNA, it would release the DNA. If it's an RNA genome, it would release the RNA. Now, of course, these guys can do two different things. They can go straight on and become infectious. Or this DNA or RNA integrates into the host chromosome and becomes latent, basically meaning it's just hanging out. So it does that by taking its DNA or RNA and sticking it into the host's chromosome, so the host doesn't really even know it. And then, of course, there is a stimulus that will make this become infectious, and we would move on to the next step. That is making all of those goody molecules necessary to make more viruses. So they co-opt the host machinery and they basically have the host make all its macromolecules. So viral proteins, viral enzymes, viral nucleic acids. So we have assembly or packaging of the entire thing together. And it's at this point that we can basically say we have a mature virus. Usually when it's inside the cell, we call it a virion, but we can call it whatever we want. And then we see release. And remember, release happens in two different ways. It can be lytic, where it just blows the cell open. The cell is destroyed. All these baby viruses go out there and attack new host cells. Or it can be actively exocytosed. And that's where we end up getting those host cell envelopes. And I had sketched that whole thing out before. We'll talk about the differences of animal viruses and those of bacteriophage and plant viruses, but I'm going to hit that a little ways down the road. To kind of back up this replication process, I think I left you guys with a viral growth curve, if I'm correct. So viral growth curves are also known as one-step growth curves. Because they're not living, they don't die. So they just go up. Once again, we're going to graph that time versus, once again, what we call virus titer. And pretty much from here on out, that's how we describe concentration. Titer can be in many different ways. It can be viruses per milliliter, parts per million, parts per billion, however we want to discuss it. Just basically the numbers of viruses. So T0, of course, is going to be inoculation. And it takes a while for that virus to find its host. In finding the host, it takes a while for it to infect. And then we get this really nice one-step growth curve. Same thing, we have a little bit bigger lag period, which we'll describe. We have this exponential. And then we just basically have a maximum population, which we describe as burst size at the very end of the growth curve. So let's start with the lag phase. Of course, a lot of different things are happening, and the actual lag or early phase, as we call it, should be broken down into a couple of different late and early sections. So sometimes this is also called the latent phase, pretty much the same thing. This is characterized by basic infection. So absorption or attaching to the host cell and penetration. And then as we enter the later part of this latent phase, we enter what's called the eclipse period. So at this point, we're getting viral enzymes being produced. Once again, my NA stands for nucleic acid, either DNA or RNA. So we have nucleic acid genome being replicated. We're starting to form those capsid proteins. And in doing so, we start to get the formation of capsids. And then we get assembly and really just initial release right at the end of all of this eclipse period. So you kind of see that initial release with just that start of the graph moving upwards. Then, of course, we enter the exponential phase, and the same as we see with bacteria. This is basically a doubling time. And that means we have complete mature virions. There's none of this biosynthesis or anything going on. And so it's really emphasized by the release step in viral replication. And then eventually we just max out at our max population, and we end up with burst size. And we can categorize burst size as the number of virions released per host replication cycle. So if I take one cell and infect it, the maximum titer I get at the end of that is what I would call a burst size. So now once again we have these kind of crazy viruses. So these viruses have the ability to have both DNA or RNA as their genome, but never both. So that doesn't mean that they don't have mRNA and DNA in the host all at the same time. It means when we have packaged this bad boy and we go to release it, and we have this nice icosahedral virus with its DNA or RNA on the inside, and it's being released, it's either going to have a DNA genome or it's going to have an RNA genome. They don't mix the types of genomes. With that being said, we have to describe some of their crazy expression replication pathways. And it depends on the type of genome they have. And so we'll kind of write this out based upon the type of nucleic acid and then the type of process that we have as far as its system. So remember, the host is doing this for the virus isn't doing this. So if you have a single-stranded DNA, and we'll just put this as genome in this column, so the type of genome that it has, that means it has just one strand of DNA. It doesn't have a double strand that goes into a helix. But what it does is it creates a double-stranded DNA helix intermediate. That then, of course, can be transcribed into mRNA. And translated into protein. Pretty straightforward, that little kind of intermediate kink in the middle there. Nothing too crazy. Of course, if it's a double-stranded DNA, it's just like the normal expression we're used to. So in this case, transcription to mRNA, translation to protein with nothing hinky in the in-between. Just like what we see with our genes. But when we head into the RNA genomes, that's when things get a little bit crazy. So we can have single-stranded RNA that we call either a positive or negative sense. And we'll kind of try to describe the difference. So we'll start with the negative sense. In this case, I'm going to put a negative behind it. It utilizes a double-stranded RNA intermediate. So this guy basically creates a double-stranded RNA intermediate. In doing so, it creates a positive strand, so we end up having both a positive and a negative strand. This positive strand basically acts just like mRNA. You don't need to make mRNA. And it basically is translated straight to protein. If you're a single-stranded RNA virus, and you have a positive strand, you don't have to make any intermediate at all. It acts just like mRNA and is translated straight to protein without any sort of in-between. And then you have these other kind of weirdos in the world. Viruses. And these are your retroviruses. And we have different types of retroviruses. We have DNA and RNA retroviruses. Most of the time you've heard of RNA retroviruses like HIV/AIDS, that sort of thing. But there are these crazy DNA retroviruses as well. So in this case, we're going to go over and put these guys down over there so I have enough room. Everyone have this top one? Good. with your retrovirus, I'm just going to put it as a retro genome. We start out with a single-stranded RNA. Well, I'll do this right away. So there are a couple of different ways this takes place, but what we end up doing is basically what we call reverse transcription. So what we do in this process we go backwards. We use this virus enzyme called reverse transcriptase. It takes a single-stranded RNA and ends up eventually making this double-stranded DNA intermediate. This intermediate then goes through the normal process of transcription and translation. And that's what we see with most normal retroviruses. And then you have these weirdos called Bunyieviridae family, and they are DNA retroviruses.
Which one was this one again?
So this one is a double-stranded DNA retrovirus. These are like Bunyieviridae retroviruses. So in this case, they have a DNA double helix, but it's incomplete. So it's kind of broken. Hasn't been finished all the way. And so these guys use reverse transcriptase to fix their double-stranded DNA. Then they go through the normal expression process.
So on the first one, it makes both RNA and DNA?
Well, it makes this duplex very quickly, and then this duplex turns in to the DNA intermediate. Most of the time, people just show you the DNA intermediate, but there is this kind of in-between time.
Okay, that makes sense.
And those are those crazy expression pathways. So one of the reasons why RNA proteins, or excuse me, RNA viruses, it's been a long day, end up mutating so quickly all has to do with the enzymes that are replicating the nucleic acids. Those would be RNA polymerase, reverse transcriptase. And what these guys are doing is they make a ton of mistakes. So they make a ton of errors during the replication. We call these low fidelity enzymes because they don't have a very good sense of when they're making mistakes. These basically turn into mutations. And so these guys are always mutating. So for instance, if we take HIV and stick it in you, by the time you die, you've probably gone through 100 iterations of that HIV virus. It's mutating, your body doesn't recognize it, then it recognizes it, the virus load goes down, then it mutates again, virus load goes up. So we see this kind of viral infectivity or pathogenicity doing a couple of different things. So we'll start with what we call viral shift and viral drift. And so viruses like to kind of add to their diversity by either recombining with other viruses. We call this virus shift. This happens through virus recombination and viral drift, just the sort of mutation we were talking about. So shift plays a large part in viral development. A good example was the influenza B virus. I think it was H5N2 or something like that. That was supposed to wipe out everyone about 3 or 4 or 5 years ago, coming through South America. Well, if you take a look at its genome is 1/3 human virus, 1/3 porcine pig virus, 1/3 avian. So this virus has the ability to jump from one reservoir to the other, and it starts to recombine with other viruses. And we see that a lot, especially in like the flu viruses and whatnot. So that's kind of our animal and expression thrown out there in a quick sense, but we need to talk about the difference between these and bacteriophage. The expression and replication pathways are the same, but their process of replication is just slightly different. So remember, these bacteriophage are using prokaryotes as hosts, bacteria and archaea. So bacteriophage are basically the most abundant entity on planet Earth and within our atmosphere. They're suggesting 10 to the 30th plus viruses, just bacteriophage viruses out there in the world alone. Same thing, they have both double strand, single strand, same sort of genomes we see in others. Single-stranded DNA, double-stranded DNA, double-stranded RNA, single-stranded RNA. We don't really see retroviruses that often in bacteriophage. That's more of an animal virus sort of evolution. So with these bacteriophage, they do basically the exact same process. Absorption or attachment. Basically, receptor mediated. Here's our big E. coli. Here's that bacteriophage coming down. Its tail fibers recognize receptors on the outside of the E. coli, and it attaches. We can actually treat this E. coli with something like rubidium chloride, and it denatures those receptors, becomes immune to the virus. So by changing those receptors, we can make them immune. Second thing, of course, isn't penetration, but rather the DNA is injected into the host. So this guy drills a little hole and it injects its DNA or nucleic acid into the host. That protein capsid shell stays on the outside of the host. It never enters. And that's one of the biggest differences that we see. So because of that, we don't need to uncoat because there's no uncoating there to do. Let's put it this way. And just like our animal viruses, they can be infectious. In bacteriophage, we call this lytic instead. Or they can go dormant or latent. We call this lysogenic instead. And likewise, same thing. Some sort of stimulus can make them become undormant and go into the lytic cycle. When we're in the lytic cycle, of course, the next thing that we see is other macromolecule synthesis. Assembly and release. And 99.9% of the time, these guys are not enveloped. So this is a lytic release, or basically lysis of the cell. They don't exocytose and grab hold of envelopes. Though even while I'm saying that, I'm sure there are these rare examples where it does happen. So a little bit different there. So with that being said, let's talk about a few of the different classes of viruses, and we're going to end off with that wonderful grand retrovirus, HIV/AIDS. I know nowadays it's not like a death sentence. When I was growing up, you got AIDS, you died pretty darn quickly. So with that, we'll kind of describe these based upon the type of viruses they are. So we haven't talked about plant viruses very much, so we'll start there and then move into the types of animal and bacteriophage. So plant viruses, same thing. They can be icosahedral, they can be filamentous, same thing. They can be DNA or RNA. Many of them are RNA. Most of these are ag nuisances. And they really don't affect other eukaryote cells whatsoever. So think of these like tomato blight. Right now, the citrus world is going crazy with viruses. Banana viruses are a really bad one as well. But what we find as an example is tobacco mosaic virus. And because tobacco was such a huge crop for so long, they would get these kind of blighted areas on the tobacco leaves caused by this virus, known as the TMV virus. This was the first RNA virus ever found. Till that point, we only thought they were DNA viruses. So it has a single-stranded positive copy. And it has helical symmetry. Meaning it is rod-shaped. And so basically it uses its genome straight to mRNA. And then we have these other things that we're going to talk about later called viroids. And these are just chunks of RNA, naked RNA, that infect plants. We'll come back and talk about those secondarily. Then also, kind of close to that plant world, we have the eukaryotes algae. And they have basically what we call chlorella viruses. These are double-stranded DNA viruses. Icosahedral symmetry or shape. And they inject their nucleic acids much like bacteriophage do. And then as we start to move up into the eukaryote world, we start to deal with those various types of animal viruses. And same thing, we'll kind of describe them based upon their attributes, their hosts, and whatnot. Let's start with animal viruses that have a positive strand, single-stranded RNA genome. And one of my favorite families is the Picornaviridae family. Same thing, they have that icosahedral symmetry. And we know these as the poliovirus, rhinovirus. I don't know if we'll get into the ideologies of these viruses. And hepatitis A are great examples. And then on the other side of this would be negative stranded RNA animal viruses. And we have a whole bunch of those. So, Rhabdoviridae is a great example of a negative RNA family. This is a huge, diverse family. And it has both plant and animal viruses in that family. These are rod-shaped or have the helical symmetry. They are enveloped, so that means that they are actively exocytosed out of the cell. Two great examples here. Of course, the rabies virus. And another one, unless you're an Aggie, you probably haven't heard of it. And that'd be vesicular stomatitis virus. We find it in ruminants like sheep, cows, and in humans as well. Still on that negative RNA sort of trend, we then head into serious human pathogens. Still negative RNA animal viruses. Orthomyxoviridae. So these guys are a little bit strange. They have multiple chromosome segmented RNA-like genomes. They have helical symmetry. They are enveloped. And within those envelopes, they have two different types of glycoproteins. And you heard me refer to them when I talked about the flu. So a good example would be the influenza virus. Both A and B strains are in the Orthomyxoviridae family. And so they also have these hemagglutinin and neuraminidase. These also mutate very quickly. So we designate these Orthomyxoviridae based upon their mutant hemagglutinin and neuraminidase. So you might have an H2N1 virus, meaning this is the H2 mutant of the hemagglutinin and the 1 mutant of the neuraminidase, or you might have an H3N5. So it's the way they describe based upon these hemagglutinin and neuraminidase spikes. Same thing, these guys undergo viral shift because we see them in a few different reservoirs. As I said before, they can jump around between birds, pigs, and humans. Now for us, we don't really hang out with our animals as much. Some of you might have some miniature pigs hanging out in your house, maybe a pigeon or something. But in a large part of the world, specifically in Asia, you'll have a house and there'll be ducks, geese, and chickens with their coop right next to the house. They have all sorts of cultures where they do things like eat cold duck blood soup. Kids will play with the heads of ducks after they've beheaded them, you know, just kind of different cultural processes. Same with pigs. And so these Orthomyxoviridae really recombine quite a bit, and that's why we see these coming out. That's one of the reasons why they're always saying your viral flu vaccine only is about a 50% chance of being on it or good. What the CDC and Health and Human Services is doing is they're trying to predict what mutant's going to show up this year. Sometimes they're completely off and they don't know. So usually your virus vaccine is a mixture of a few different mutants, a few different viruses all in one go. So you have about a 50-50 chance that it's going to work, 50-50 chance it's not going to work. By the way, something that's not really talked about very much, on an average year we have right around 10,000 influenza deaths. Isn't that pretty incredible? We don't really hear much about it. How many cases of Ebola have we had in America? 2. God, that was all over the news for like months on end. Thousands and thousands of people that die every year from just these very common type of viruses that we see. Let's move into the double-stranded RNA viruses. Once again, we're still in the animal world. Likewise, kind of like we see with the Orthomyxoviridae, they have segmented chromosome-like double-stranded RNA genomes. These are usually icosahedral in shape. They're normally enveloped. Oh, excuse me, non-enveloped. And they are strange in they have a double-walled capsid, or a double shell. So kind of a capsid shell inside of a capsid shell. So this makes them super tenacious and able to hang out in the environment quite a bit. So we think of those as being environmentally stable. Some viruses don't do well outside of their hosts. They just don't have the ability to take all the stresses of the environment, radiation, heat, drought, all of that. And the reason why these guys are this way is because they belong to the Rhinoviridae family. So think of these like the Norwalk or can't remember what else they call it, Norwalk-like viruses. So these are your gastroenteritis GI viruses. So that means they are completely past fecal oral. So that means they come in through the mouth because you're eating poop. They make it through your digestive system, create something like 12 to 48 hours of horrible diarrhea, vomiting, all of that. Then they shed out into the environment and they wait for the next stooge to grab them and eat them again. So they have to have this pretty stable double-walled capsid. And one of the last ones we'll leave it on today is that wonderful Poxviridae family. So we're moving into the DNA world. We'll go straight to those normal double-stranded DNA animal viruses. And we'll go to the Poxviridae. So we find these are usually one of the most complex shapes that we know of. Most of these are icosahedral. Most of them are enveloped. That means that they enter through endocytosis, exit through exocytosis. And so they're some of the largest viruses that we see on Earth. And so a good example of this is the vaccinia virus, also known as smallpox, one of the most infectious viruses we know on Earth besides the measles. And it is more or less has been eradicated from the human reservoir. In saying that, pretty much all countries that have a bioweapons system or bioweapons program has smallpox and smallpox vaccines out there. So it could make a reemergence at any given time. We also see other types of pox, cowpox, etc. And we'll leave it there for tonight. Before we go, however, let me show you something really quickly. Oh, sorry about that. Head down to lab.
Did you not jump? That's the question.
What do you think, guys? Little virus porn. Yeah, no, this is actually mine. I'm just kidding. So here we have kind of what we're talking about in these shapes and sizes. So this is a true scanning electron micrograph of a icosahedral virus. So you can see those individual capsimers that make up the entire capsid. And just kind of showing you that whole process where we see the capsimers and the capsid all coming together. And the difference between just a plain, what we would call just a encapsulate, or excuse me, a capsid virus, and then that which also has the post envelope around it. So here's an example of the different types of shapes. Now recognize this is one of the few self-assembly processes in nature. Just because of the geometry and the biochemistry of these capsimeres, they kind of stitch themselves together. It's really a pretty crazy process. But you can see, even though we call them icosahedral, they all have a little bit of different variations. And believe it or not, these various proteins form patterns on the outsides of these. This isn't a great picture to show that. There are others. So this is that icosahedral shape. Now when we're talking about the rod shape, the filamentous shape, they're a little bit different. Their capsids and their viruses are a little bit weird. So the RNA kind of coils, and then those capsids, which make up the capsimere, just kind of wrap around that RNA until you get this nice filamentous virus. And it's just showing you the difference between an icosahedral, filamentous, how they can both be enveloped. Now, as far as bacteriophage, one of the ways that we can play with bacteriophage is just to grow a whole lawn of E. coli and then drop the virus onto the E. coli. And we can look for spots where the E. coli are just lysing. And this is kind of what we're seeing right here. These are called lysis plaques. So each one of these plaques is where a little bacteriophage virus has been dropped onto the E. coli. And slowly but surely, it's just busting those E. coli up and lysing them. We also see this with animal viruses. If we do like a layer of cells, animal cells, we'll get these same sort of animal cell plaques as well. Just a process showing you how these bacteriophage kind of use various types of enzymes, specifically lysozyme, to kind of cut through the various types of molecules. They're able to inject their DNA down in there. Just showing you the differences in their size, a bunch of different variations on it, how this happens at the cell wall. And here we have different kind of electron micrographs. So this is what we call the Marburg virus. These are what we call Bunyieviridae. So Marburg, Ebola, a bunch of other hemorrhagic viruses, specifically out of Asia, where we first found them in Vietnam and Korea both. So this is kind of an electron micrograph. It kind of always form these kind of number 9 looking shapes eventually. Different from Ebola. Ebola has this really nice cobra looking look to it. So this is a filamentous virus, but it kind of folds around on itself. Here we have influenza A. You can see on the outside all of those hemagglutinin and neuraminidase spikes kind of shooting out of the envelope there. I think this is the lacrosse virus. And here's an example of those patterns I'm talking about. This is actually an electron micrograph, but it's been enhanced with computer color. This is the Zika virus. And you can kind of see these kind of swirls coming around, starting to swirl in those proteins. So they have these really complex shapes to them. Pretty cool. And that's it for now. I will see you guys down at the quarter after, and we'll finish up our staff and be done for spring break.